Myself, Dr. Shilpa Bhosle, Assistant Professor, Dempe College of Arts and Science, Miramar, Goa. Program, Bachelor of Science, first year. Subject, Botany. Semester 1, Course Code BOC 101. Course title, Biodiversity 1, Microbes, Algae, Fungi and Bryophytes. Unit title is Bryophytes. Module name, Reproduction in Anthoceros. Sexual. The outline of the module is the concept of sexual reproduction in Anthoceros. Learning outcomes. The students will able to explain the concept of sexual reproduction in Anthoceros. Coming to introduction. Reproduction, it reproduces vegetatively and sexually. Asexual reproduction is absent in Anthoceros. In the picture, here you can see the Anthoceros habit where you can see the thallus, the involucre and the sporophyte. The hand-drawn picture also shows the thallus, the involucre and the mature sporophyte. Sexual reproduction. In Anthoceros, sexual reproduction is oogamous. That is, the reproduction by the union of mobile male and immobile female gametes. Here, the species may be monoecious, that is, having both the male and the female reproductive organs in the same individual. Or sometimes dioecious, having the male reproductive organ in one individual and the female in another. Monoecious species are protoandrous, that is, antheridia formed before archegonium. Sex organs are formed endogenously embedded in the thallus. Coming to antheridia, they are formed singly or in groups on dorsal side in the antheridial chamber. Each antheridium bears a stalk which is multicellular, formed of a row of cells. The body is oval and consists of antheridial wall, that is the jacket wall enclosing the anthrocytes. When mature, antheridias are red in color. The picture showing the mature antheridium inside the antheridial chamber. Here you can see on the stalk the antheridial cells are enclosed with the jacket in the antheridial chamber. Anthrocytes develop into an anthrozoid or the sperms. Each sperm is tiny, biflagellate and having flagella of equal length. In the picture, you can see the mature sperm having the biflagellate flagella. They are isoconic type. At maturity, the roof of the chamber breaks out, followed by the opening of the jacket through which the anthrozoids are liberated. In this picture, you can see the liberation of the anthrozoids where the roof is broken and the liberation of the anthrocyte mass is occurring in the antheridium chamber. This is the structure of the mature archegonium. In the mature archegonium, you can see the egg cell, the ventral canal cell, the neck canal cells and the cover cells. Coming to the archegonium, these are formed on the dorsal surface sunk deep into the tissue of the gametophyte. Formed in row in an acropatal succession, that is, proceeding from the base towards the apex or from below upward, that is the acropatal succession. The cover initials divides to form the distal rosette or four cover or the lid cells. Each archegonium have 6 neck canal cells and 1 ventral canal cell and an egg. Sterile jacket layer around the axial cell is absent except cell at the apex, that is the lid or the cover cells. Cover cells slightly projects above the surface of the thallus. Coming to the fertilization, on the maturity of the archegonium, the neck canal cell and the ventral canal cell disintegrate, forming mucilage. Here, due to the pressure of this mucilage, cover cell opens out, and here sperms are attracted to the mucilage. One anthrazoids unite with an egg cell, forming a zygote. Number of zygotes are formed in a single thallus, and this is the first cell of the sporophytic generation. These are the reference for the module. Thank you.